students, we are in the second video of Biotechnology Principles and Processes chapter. In the first video, we looked at the steps that are usually taken to extract a product or to make it in large scale finally. So the entire steps that we usually go through for biotechnology, we went through. In that, in our textbook, if you notice, this is not the sequence in which it is given. How are they given? They have divided all this into tools and processes. Okay, tools on one side and then processes on the other side. Do not worry, I'll be teaching in this sequence and I'll be telling you which among them are used as tools and the other one, which all will come under the processes part. Okay, we'll be going in this sequence, however. All right, so first and foremost, we identified our gene of interest. That is self-explanatory. I don't think we need to go into that anymore. If you haven't understood, please go back and watch the first video. All right, so second thing, after identification of your gene of interest, what do we have to do? We need to extract the DNA out of the entire organism, at least few cells of the entire organism, so that we can take out our gene of interest from the DNA. So this, I have taken some pictures of a plant cell and an animal cell just to give you an idea of what we're talking about. So here is your DNA. In the nucleus is your DNA. This is a plant cell, this is your animal cell. In both cases, we have our DNA. In the DNA, there is one part which is of interest to us. However, we are going to take out the entire thing and then isolate our gene. All right. So, to extract our DNA, what do we have to do? We need to use certain enzymes to cut through all these layers so that finally we get only DNA. So, in a cell, as you know, it is not just DNA that is present. In a cell, there has to be a lot of RNA, there has to be a lot of proteins, lipids, polysaccharides, and so on. Correct? So, what do we have to do? We need to cut through all these and get rid of everything else that we don't want, precipitate it out using centrifugation or some other method, and take out only our DNA. How to do that? All right. So, first and foremost, we will look at all the organisms that has one extra layer of barrier, like for example, plants. What layer am I talking about? I'm talking about the cell wall. Plants, fungi, bacteria. All these have cell wall which needs to be initially taken out. Only then we can enter inside the cell and take out our DNA. Right? We are extracting the DNA. Okay. So for plants, fungi and bacteria, different enzymes are being used. For plants, the cell wall, since it is made up of cellulose, we will use the enzyme cellulase to get rid of it. In fungi, since the cell wall is made up of chitin, we will use the enzyme chitinase. And in case of bacteria, the cell wall is made up of peptidoglycan, which can be broken only by this enzyme called as lysozyme. Once this set of enzymes, either one of it is added, depending on the depending on the organism from which we are extracting the DNA, depending on the organism from which we are extracting our DNA, in which our gene of interest is present, depending on that, first step is addition of any of these enzymes. Done? So we've gotten rid of that. Now inside the cell, there is going to be a lot of RNA, a lot of cell organelles. All those could be made up of proteins, lipids, polysaccharides, and so on. So what do we add next? We add protease. Protease for what purpose? To get rid of all the proteins, especially in case of plasma membrane, a lot of proteins are going to be present. Then what do? We need to get rid of the RNA that is there in the cell. So for that, we add RNase enzyme. So all these are added. Lot more processes are also to be done. However, it is not mentioned in our NCERT, so we are skipping. Then once you get the DNA, which is purified to a larger extent. How do we get this out? How do we precipitate it out? We precipitate it out by using chilled ethanol. It is clear? We'll continue in the next video.